What is plant virus assay? Is it different from detection and diagnosis techniques? So in this video we are going to discuss about various methods of virus assay, and we will also discuss how it is differ from detection and diagnosis techniques. So first about the virus assay. So virus assay means quantification or quantitative estimation of virus concentration, infectivity or the biological activity of a virus in the host plant. But in contrast to this, detection and diagnosis means, if you say the detection means we will detect what pathogen it is, whether it is a virus, bacteria or fungi. So we will detect whether the pathogen is a virus or not. But diagnosis means we will diagnose which virus it is. Okay. So detection and diagnosis means we will check whether the pathogen is a virus or not. If it is a virus, then which virus it is. And the virus assay, it is little advanced than detection and diagnostic methods. So in the virus assay, we will do the quantitative estimation about the concentration and infectivity. So the major objective of virus assay is to determine the virus concentration associated with the host or any samples, then measure the infectivity. So how many infective variants are there that is causing the disease? So it also measures the infectivity and estimate the viral components. What are the components are there? We will estimate that. Then the quantify the viral genome. Virus concentration means we will quantify the genome only. Virus is just a simple nucleoprotein structure. Okay. Then about the classification of virus assay. So virus assay can be categorized into 5 types: biological assay, second physical assay, third chemical assay, fourth serological assay, and fifth molecular assay. Similar classification is also there for detection and diagnosis technique, like the conventional and the modern biotechnological techniques for detection and diagnosis of viruses. So we'll discuss this in the later videos. Now in this video we will focus on virus assay, how to quantify the virus and how to quantify its infectivity as well. So next, first about the biological assay. So biological assay, the principle behind this assay is it is based on the infectivity of virus and symptom expression on the host plant. So in biological assay, we will check the infectivity. Major concern is infectivity. Based on infectivity, we will quantify the virus. So methods included are local lesion assay, infectivity assay, dilution endpoint assay, and host range assay. First one is the local lesion assay. This is the most important assay. This is the first assay used in the plant virus for the estimation of concentration, and it was developed by Holmes, Holmes, Holmes 1929. This is a very important MCQ based question, repeatedly asked in ASRB NET or other competitive exams. So local lesion assay was developed by Holmes in 1929. So what we use? We use the indicator host plant. So indicator host plant are nothing but are the special kind of plants in which that particular virus, when it will be inoculated, that will produce some specific symptom. Okay, so what happened? The plant will produce local lesions that will directly, actually, the local lesion number will be directly proportional to the virus concentration. If we count the number of lesions formed by or formed after artificial inoculation of the virus, then we can predict the virus concentration. More the number of local lesions, more will be the virus concentration or virus titer. So generally for concentration, titer term is used for the virus titer. Okay. Example of indicator host plant are Chenopodium amaranticolor, Nicotiana glutinosa. This is for TMV, okay, tobacco mosaic virus. So for all viruses, they have their specific indicator host. So we will deal with it in later videos, okay. Then next, second important method or the biological method is dilution endpoint assay. In the physiological properties, I have already discussed that dilution endpoint means the highest dilation, after which when we will inoculate that sap on plant, it will not produce any symptom. So that is the highest dilution or dilution endpoint. So each dilution is inoculated on a suitable host, and highest dilution showing infection and symptom is recorded as endpoint. So for various virus, means for different virus, the dilution endpoint is different. So based on that, we can check that. For the TMB, it is 10 to the power minus 6. Okay. Next, the infectivity assay. So to know the dilution of virus, sap is inoculated on the test host plant and percentage of infected plant is calculated. So here in the biological assay, what we will do? We are checking the infectivity. So here in infectivity assay, the infectivity percentage is calculated by the number of plant infected by total number of plant inoculated. Simple method, okay. Next, the host range assay. Simple like the infectivity assay. We will just inoculate the virus, the artificially collected, isolated, that sap will be artificially inoculated over the host plant. And all the viruses are very specific. Virus show very high host specificity as well as tissue specificity. So based on its symptom expression or based on its ability to infect host, we can predict we can study the infectivity on, and also we can do the biological assay. So this is the host range assay. So this is about the biological assay, means how it is performing or producing symptom in the host. So based on the expression of symptom, we are assaying the virus infectivity and virus concentration or titer. Then about the physical assay. Physical assay is based on the physical properties of virus. So we will check about the protein structure as well as the nucleic acid structure. So what are the methods that included in the physical assay? They are, first, the most important, UV spectroscopy. As you all know that the nucleic acid as well as protein, they will absorb different spectrum of UV light. So this nucleic acid generally absorb UV light maximally at 260 nm, whereas the protein absorb UV light maximally at 280 nm. This can be directly asked in the MCQ based question. Okay, remember this. For nucleic acid, it is 260 nm, and for protein it is 280 nm. Therefore, 260 by 280 ratio is actually used for purity assessment. By this ratio we can quantify the proteins as well as nucleic acid. So for pure DNA, this ratio actually ranges is the ratio actually approximately 1.8. For pure RNA, it is 2. So generally, actually, we use some nanospectrometer. Generally, for RNA or DNA study, nanospectrophotometer we use. The instrument name is nanospect. Normal spectrophotometer we generally use, as the DNA and RNA are very smaller in quantity. So we use nanospectrophotometer, and then after putting the sample, we will check this ratio in this device itself. It will show the ratio. So if that ratio is around 1.8, we will call it that as this is a pure DNA. It is not contaminated with proteins and other things. And if this ratio is around 2, we will call it as pure RNA. Okay. So here we are assaying the nucleic acid as well as protein. So nucleic acid means if it is a pure RNA, if we are extracting a virus particle, and if we are getting pure RNA, means it is a RNA virus. If we are getting the DNA, means it is a DNA virus. Like that we can predict. Next, second important thing is electron microscopy. So electron microscope, in microscope what we use to see? We use the electron microscopy or any microscope to see the shape, size, physical appearance we used to see. So similarly, electron microscopy is more advanced version of microscopy, where we used to visualize the virus, and virus can be only visualized using the transmission electron microscopy, TEM. So actually two types of electron microscopy are there: scanning electron microscopy and transmission electron microscopy. But with the TEM only, transmission electron microscopy only, the virus can be visualized, not with the SEM, scanning electron microscopy. But and other type of normal microscopy, we can't visualize the virus. So by using the transmission electron microscopy, we can count the virus. Okay. So various information about shape, size, morphology can be detected, or information can be generated. Next, the most important thing, ultracentrifugation, analytical ultracentrifugation. This is the advanced version of ultracentrifugation. So centrifugation, as you all know, that the particles will be separated based on the sedimentation rate in a centrifugal field. So for virus, I have already discussed what is the sedimentation coefficient for the virus. That is ranges from 10 to 1000 S. For CMV it is 200 S. Okay. For different virus, this sedimentation coefficient value is different. So by that we can estimate the virus. Then about the light scattering method. So this is actually the confocal microscopy. Okay. By scattering of light, we can, means the intensity of light actually directly proportional to the concentration of the particle. So it includes nephrometry or turbidometry method. So virus, by this we can predict, means if the more viruses are there, more scattering will be there. So by this we can predict the concentration. So 4 methods are there for physical assay: UV spectrometry, electron microscopy, analytical ultracentrifugation, light, and fourth one is light scattering method. Next, about the chemical assay. So in here we will do the quantification of viral chemical constituent. So as you all know that virus is chemically nucleoprotein structure, nucleic acid and protein. And apart from this, some other molecules, like, because it's a biological thing. So definitely nitrogen, phosphorus, carbon, all the things are also there. So the chemical methods, the principle behind this is estimation of either protein or nucleic acid, or by nitrogen and phosphorus analysis. So 3 methods are there. So the first method is protein estimation. If anybody is working in molecular biological laboratory, they might heard about this assay name, Lorry method, Bradford assay, biuret assay. So these are nothing but the protein estimation assay, very standard method for estimation of or quantification of protein. Okay, no need to study in detail. They are very complicated methods actually. So generally you can just remember this, that this Lowry method, Bradford assay, biuret assay, these are method for protein estimation to measure the total protein content in a virus, because for viruses the protein content is different. Okay, so by that we can predict the virus, and means if more protein is there, more virus will be there. So concentration also can be assayed or can be estimated. Next, similarly, we can measure the nucleic acid. So nucleic acid, how we can measure? Similarly, we can use the spectrophotometric method. I have already told, if it is a pure RNA, the ratio will be around 2. If it is pure DNA, the ratio 260 by 280 ratio will be 1.8. So this 260 or and 260 by 280 nm, this ratio is used for the nucleic acid estimation. Next, the phosphorus and nitrogen analysis. So here the nitrogen phosphorus content in the purified virus preparation was estimated. So this actually, nitrogen gives the indirect information about the protein, and this phosphorus gives the indirect information about nucleic acid, because phosphate group is there for nucleic acid and nitrogen is of the major constituent of proteins, amines. Okay, so this nitrogen estimation done by Zeldal method in the Zeldal tube, test tube, not tube, Zeldal, that flask is there. It is in the actually, if you study in detail about the laboratory method for estimation of nitrogen in soil science, you can see the Zeldal method. Zeldal flask is there, by using which this nitrogen estimation was done. And similarly, this phosphorus also, it is actually colorimetrically analyzed. So this method actually we have already studied in our UG in soil science. Okay, so this nitrogen actually indirectly estimate the viral protein, and this phosphorus indirectly estimate the viral nucleic acid. So this is about the chemical assay. So chemical assay means we are estimating the either the nucleic acid as well as the protein, because the major chemical constituent of the virus is nucleoprotein. By that we can estimate the virus concentration, infectivity. Okay, next, about the final assay, molecular assay. So molecular assay means we will detect the molecules, that is the nucleic acid, DNA or RNA, using advanced technique. So advanced techniques are either it may be PCR based, okay, either it is PCR based, or it is hybridization based, or it is advanced assay. So the PCR or amplification based assay are, amplification means we will multiply the DNA or RNA into million times. Okay, so the methods used are very common method: PCR polymerase chain reaction, or RT-PCR, or real-time PCR, or digital PCR, or LAMP, loop mediated isothermal assay. See, this is a very important thing. Loop mediated isothermal assay, how it is different from the normal PCRs. These are PCR, but this is not the PCR. How? Polymerase chain reaction. So this is a chain reaction are there. At different temperature, different reaction are performed to amplify the particular nucleic acid. But loop mediated isothermal acid. Isothermal means at a constant temperature this amplification is going on. generally at 60 to 65 degrees Celsius. So this is isothermal based method, whereas these are polymerase chain reaction. This is the difference between these two methods. Next, about the hybridization method. So in hybridization method, complementary sequence will be hybridized. Either DNA-DNA hybridization or RNA-RNA hybridization will be there. So the method include are dot plot, southern blot, northern blot. Northern blot for RNA virus, southern blot used for DNA virus, and this dot plot actually in a membrane, first nucleic acid will be fixed, and that over that the labeled probes will be incorporated, and we will check the hybridization status. So these method we will study in detail in laboratory techniques one by one. Okay, one by one we will study in detail how this, each and every step we will discuss. Then about the advanced technique, you might heard about this NGS, next generation sequencing. So various sequencing techniques can be used for detection and or quantification of the virus nucleic acid. And among these methods, the most advanced as well as most highly sensitive as well as specific technique for plant virus assay is this advanced technique NGS. Highly specific, highly sensitive, rapid, very useful. Okay, so this is all about the virus assay methods. This is the comparison table. So 5 methods we have already discussed: biological assay, physical, chemical, serological, and the molecular. So serological, I think I forgot. Serological, one second. Oh, I forgot to put the slide. So serological is nothing but, so in serological method, serology means it is based on immunoprecipitation methods. So immunological methods. Here actually the base or principle is it is based on antibody and antigen interaction. So these two are antigen and antibody are two proteins. So these proteins will interact. They will form a precipitation. If it is forming a precipitation, it will result means they are interacting. If it is not forming any precipitation, means it is not interacting. So by the result actually we will detect whether the virus is there or not, and also the quantity as well. So various methods included in the serological methods or serological assay are ELISA, immunodiffusion, immunoelectrophoresis, latex agglutination, immunofluorescence assay. So we will deal with each and every assay in detail. Okay, what is ELISA, types of ELISA, direct ELISA, indirect ELISA, sandwich ELISA. Then about the conventional ELISA, conventional this serological method, and advanced method. Means in advanced actually this lateral flow assay. These things are there. So very complicated things we will discuss one by one. Okay. So this is the comparative table. So in biological assay, what we do? We study the symptom and expression of the virus in the host plant. So indicator host is used. Method include local lesion assay, dilution endpoint assay, infectivity assay, and host range assay. But this is a very time consuming method because we have to inoculate the virus, then we will have to wait for the symptom expression, then we have to quantify. So this is very complicated, means time consuming. It is also influenced by the host factor as well, and it is less sensitive. Then about the physical assay. Physical assay is based on physical properties of the virus particles using spectrophotometry, then electron microscopy, analytical ultracentrifugation, and light scattering method. So the virus particle shape, size, these things are estimated. And limitation are, it requires specialized equipment like transmission electron microscopy, ultracentrifugation. So specialized equipment it required, and it just assess the shape and size. It cannot distinguish the infective and non-infective particle. This is a major limitation of physical assay. Then about the chemical assay, assay based on the nucleic acid as well as the protein. So for protein estimation, this Lowry Bradford Biot assay methods are used, and for nucleic acid estimation, generally this 260 by 280 nm ratio is used, which is estimated using the spectrophotometer. And nitrogen and phosphorus analysis I have already told. Nitrogen analysis actually it indicate or indirectly estimate the protein by Zeldal method, but this phosphorus analysis we do for indirect analysis of nucleic acid by the colorimetry method. Okay, so major limitation is it doesn't indicate the infectivity, only the chemical composition it will indicate. Then serological method I have already told. Then finally the molecular assay. These are the most advanced assay, most sensitive assay, but only limitation is it require costly equipment as well as costly chemicals. Okay, so technical expertise is there to handle the lamp. Next, NGS. The most important technique is the NGS. Okay, so this is all about the virus assay. How assay means how this virus means in the plant sample or in the host sample the virus is present. Means we have already detected the virus. Now it's time to detect at what concentration the virus is there and about the infectivity status, how much infective it is. Okay, so in the next video we will discuss about the comparative table for the virus assay and detection, and finally we will discuss about the conventional as well as the biotechnological methods for detection and diagnosis of plant viruses. Okay, this is about today's video. I think you got the point about virus assay methods, and I think it will be helpful for you for various exams. So thank you for watching this video, and don't forget to like and subscribe my channel.